Hi, I'm Beatrix Suess and I will talk about engineered riboswitches. RNA is a very interesting and very fascinating molecule. Due to its internal flexibility, it can adapt different conformation or alternative structures. And nature used this for control of gene expression. And these are the so-called riboswitches. Riboswitches are structured RNA elements which are located in the 5' untranslated region of many bacterial messenger RNAs. And they are composed of two domains. The Aptamer domain in orange can recognize the ligand with very high affinity and specificity. And we have a second domain, the expression platform. And the expression platform interprets the binding state of the aptamer and leads to the regulation of gene expression, either by transcription termination or by the control of translation initiation. There are several advantages of riboswitch control. We have a direct interaction between the ligand and the RNA with a very high affinity and specificity. And the RNA here is a sensor, but also the regulator. So we do not need further cofactors for regulation. Due to the molecular nature of RNA, you can construct even more complex RNA regulators. So this predestined this mechanism or this principal riboswitch mechanism for to build up engineered riboswitches for the conditional control of gene expression. And another advantage of RNA is that you can build up such binding or such sensing domains, the so-called aptamers, de novo by a process called in vitro selection or SELEX. And this method I want to introduce you here on the next slide. You start with an initial RNA pool, which is a very huge library of different RNA species, let's say 10 to the power of 14 different RNAs. And then you have your target of choice, and you immobilize your target, and then you incubate the RNA with your target, and you will remove all the RNAs which cannot bind to your target. But a few of them have the ability to recognize your target, and you can specifically elute them and then reverse transcribe it back into DNA. You can amplify the DNA, and this DNA you can then in vitro transcribe back into your RNA, and then you end up here after the first cycle, and then you can repeat this selection process several times. And if you are lucky, after, let's say, 5, 6, 7, 10, or 15 rounds of selection, you will end up with small RNAs which can recognize your ligand of choice with a very high affinity or specificity. In principle, you can make this procedure against a multitude of targets. So you can find aptamers against small molecules, against peptides, proteins, or even against whole cells. And finally, you will end up with such small guys here, which in this case recognize the drug tetracycline. Such aptamers are characterized, as already said, by a very high binding affinity and specificity, and they have also, or some of them, undergo structural changes upon ligand binding, and this we wanted to use to build up our synthetic riboswitches. The principal idea is shown here. In the upper part, you see that we have integrated an aptamer into the 5' UTR of a eukaryotic messenger RNA. And without a ligand, the aptamer is only pre-structured, only slightly structured. And then if you add the ligand, you, here in this case, the aptamer adapts the defined tertiary structure, and in this conformation, it's able to interfere with cellular processes. Here, in this case, it can stop the scanning ribozyme, and this can't recognize the start codon, and so it leads to the stop of translation. We started our work with an aptamer which can bind tetracycline. As you can see on the left side, this H-shaped RNA structure, it forms a very complex structure here. And in green, you see tetracycline, which is deeply buried within the RNA molecules, and nearly all chemical moiety of tetracycline are read out by the RNA. So it's a very tight complex. And this aptamer we inserted in front of a GFP reporter gene in yeast. And here you can see that with increasing concentration of tetracycline in the broth, we obtain a decrease in the GFP fluorescence. The control is very tight. If you insert more than one, but 3 copies of this aptamer, then you obtain really a tight regulation of gene expression. You can even use this to control essential genes. Here it's exemplarily shown for NOP14. It's a protein involved in ribosomal biogenesis. And if you bring this under control of our tetracycline aptamer, and if you add tetracycline, you can completely inhibit the growth of tetracycline as it's shown here. It's a very rapid decrease, so you can't detect the protein anymore after 4 hours. Beside the control of translation initiation, we also wanted to control splicing. And again, we started in yeast, and the idea is schematically shown here. Now we have an intron which is inserted in the GFP reading frame of yeast. And then within this intron, we inserted our small aptamer close to the 5' splice site, which is shown there as a red box. And the idea is that the 5' splice site is accessible without tetracycline, so the spliceosome can bind. And then if you add tetracycline, you have a tightening of the complex, and the 5' splice site is not accessible anymore. Here you can show some of the constructs. We have inserted the aptamer 2 nucleotides away from the 5' splice site. This is the left construct, directly behind the splice site. And then we also have integrated the 5' splice site into the stem of the aptamer. And what you can see is that as soon as you are very close on the splice site, then you can nicely inhibit gene expression, as you can see on the gray bars. And the best construct we have if the splice site is completely integrated within the closing stem. And we think that there is a certain breathing of this closing stem, and after binding of tetracycline, you inhibit this breathing, and then the splice site is not accessible any longer. And here you can see that you indeed, on the level of RNA, can distinguish between the construct without and plus tetracycline. A next cellular target, besides translation initiation and splicing, was mRNA degradation we wanted to control. Here we made use of small RNA molecules, so-called ribozymes, and these ribozymes can cleave itself. So if you integrate such a ribozyme into an mRNA and it cleaves itself, then the RNA is rapidly degraded, as you can see on the left side of this picture. But we wanted to control the cleavage activity by an aptamer, shown on the right side, so that we can regulate if the mRNA is degraded or not. Here, I want to show you our construct. This is the secondary structure and the tertiary structure on the left of the hammerhead ribozyme. Only if the ribozyme folds into the structure you can see on the left side, then it cleaves itself exactly on the position which is marked by the red arrow. And then we attached our tetracycline aptamer on the right side. And very important for the activity of the ribozyme is the loop-loop interaction, which is shown here between this and the loop on the other side. And we thought maybe we can disturb this loop-loop interaction by our tetracycline aptamer in a way that the nucleotides which are encircled are free and are there for loop-loop interaction if the closing stem of the aptamer here is somehow breathing and not rigid. And then if we add tetracycline, as shown in the last experiment, there is a tightening of this closing stem, and these 3 nucleotides here are not longer accessible for this loop-loop interaction, hence the ribozyme can't cleave any longer. And so we looked for some closing stems which exactly behaved in this way. Here you can see some constructs which we tested, and indeed there we found 3 stem compositions which showed exactly the behavior we wanted to see. Here, without tetracycline, you have this breathing in the stem, and you can, this allows the ribozyme to form, and you have the mRNA degradation. That means without tetracycline, these are here, is shown here. Without tetracycline, you have no gene expression, and then if you add tetracycline, you prevent the breathing of the closing stem, and the loop-loop interaction is disturbed, and this leads to stabilization of the mRNA and to an increase in gene expression. We tested this not only in yeast, but also in eukaryotic cells, in HeLa cells. Without tetracycline, no fluorescence. If you add tetracycline, then you have an increase in reporter activity. To sum up this first part, I hope I could convince you that you indeed can use such aptamers to control gene expression. You can use a tetracycline aptamers. There are some others known in the literature which bind theophylline or neomycin. You can work in different organisms, bacteria, yeast, archaea, human cells, and you can target different cellular targets. So, for instance, the translation initiation, you can control splicing or mRNA stability. However, if you have a look into the literature, you realize that these 3 aptamers, the tetracycline, neomycin, and theophylline aptamer, these are the only ones which are used to build up riboswitches. And that's why we were interested in the question, what turns an aptamer into a riboswitch? And therefore, we try to find further of these guys, further of this regulating aptamer, this time the selection was done with neomycin as a target. After several rounds of selection, we ended up with an enriched pool of aptamers. So this means within these pools, there are several aptamers which can bind theoretically to neomycin. But then we did not continue with this pool and sequence it and looked for single colonies, but we used the complete pool and we cloned the pool in front of GFB reporter genes. And then we screened on 96 well plates for aptamers, which have exactly the phenotype we looked for. So here we have found one aptamer with, if we have it inserted in front of a GFB reported gene, led to decreased fluorescence if we add neomycin. The sequence is shown here. However, if you have a look into the enriched aptamer pool here, then this aptamer behaves similar to the tetracycline aptamer, and it leads to a nice dose-dependent decrease in gene expression if you add neomycin. However, if you sequence this in vitro selected aptamer pool, and this is the red guy here, then you end up with a completely different aptamer. You have the same stem loop structure, but the sequence is completely different. That's why we were interested why the one guy is active, the black one, and the red one was not able to for build-up riboswitches. And so we characterized this active aptamer, and we could show that the upper part is important for the binding of the ligand. However, we could identify here a small element, we call it asymmetric bulge, so some nucleotides on the left side of the aptamer, which we identified as important for regulation. So this bulge can be between 1 and 5 nucleotides long. It's mostly sequence independent with a small preference for pyrimidines. However, it's important that it is present, and it's important that you do not have a nucleotide on the other side of this bulge. At this point, we decided to team up with the NMR group in Frankfurt, the group of Jens Wöhnert, and he analyzed the structure of this aptamer for us. And he could find out that the aptamer is largely unstructured in the absence of neomycin. You only can identify the 7 Watson-Crick base pairs which are shown in the secondary structure. However, if you add then neomycin, you can see in the lower spectrum a lot of new signals, these red dots, and they correspond to several new base pairs. Here we have 2 UU base pairs. The lower ones is hydrogen mediated, so it nicely fits into an A-form helix, and this allowed the adjacent GC base pair to form. And then the upper and the lower part of this small aptamer stack on each other so that you have a long, rigid helix. And this helix then is able to interfere with cellular processes. So if you compare our active aptamer here, then you have this bulge, and this bulge seems to keep open your ground state, and this is not the case for the inactive aptamers. As you can see on the left side, these are similar aptamers. However, in this case, they are non-active in vivo, and they also do not show any conformational changes. So we think, or after a further analysis of this mechanism, we could show that we have in the ligand-free state a kind of dynamic ensemble of a kind of open and a closed state in an equilibrium, and then the ligand comes and binds the preformed closed state and stabilizes like a clamp, which holds the upper and the lower part of this helix together. And this is not a case for the inactive aptamer, because there we have already the major population of this closed conformation and no destabilized population. I want to summarize my talk. I hope I could convince you that you can indeed use small molecule binding aptamers to control gene expression, to engineer riboswitches either by using the aptamers alone or by building up more modular riboswitches. You can control different cellular processes like translation initiation, splicing, or mRNA stability, and you can build them up for different organisms. And in the second part, I gave you some mechanistic insights into these engineered riboswitches. I showed you some experiments for a Neomycin Aptamer where large conformational changes occur, and that these large conformational changes are important for the regulatory activity of this aptamer. And very important, the active aptamer was completely underrepresented in the pool after in vitro selection, and it was important to add an in vivo screening step to identify this regulating aptamer. Thank you very much.